So here is the live stream link. In case you want to have it, you do not have to use it for anything. Um, um, all right. So hello, everybody. My name is Tara, and I am the National Media Chair for NeuroSci. My pronouns are they, she, and it is my pleasure to welcome you all to our day two. The slide is wrong, but it's day two of our grant recipient panel with our fabulous panelists. We will do like a little breakdown of the schedule and um, now. And the first will be going over some grant and award dates, since that is kind of what we're all talking about a little bit here. And then I'll introduce our panelists. And for at any point, if you have any questions, feel free to put them in the chat, either on Zoom or on our YouTube, and they'll get answered at any point. We'll have a short little break, and then we'll just kind of open it up to a larger conversation. So thank you so much for being here. Our award and grant dates. The first one is coming up next week on Friday, and that is our undergraduate research grant, which is up to $1,000 for a research project. And then our outstanding student member of the year, outstanding faculty member of the year, and chapter of the year awards. Those applications are due on Friday, May 2nd. So still some time for those. And now to get into introducing our panelists. Our first panelist is Alexander Trendov, who is a current master's student in the basic medical science master's program at Wayne State University, having previously obtained his BS in neuroscience with a minor in business administration from Wayne State University. After the completion of his master's degree in May 2026, Trendov plans to attend either an MD or DO medical school program and currently hopes to continue his training after medical school as a neurologist. As a master's student, he is focusing on clinical work, prioritizing his master's classes, and preparing for the medical school application cycle. Trendov was awarded a NeuroSci travel grant for his project, Preclinical Model of Palizaeus Merzbacher Disease, an in vivo study, under the guidance of his former PI, Dr. Alexander Gao. The Gao Lab focuses on myelin research with an emphasis on neurodegenerative diseases, utilizing a variety of techniques, and Trendov specifically focused on the study of PMD. In the travel grant study, he utilized an AAV vector containing a short hairpin RNA sequence as a therapy to mitigate disease symptoms, the efficacy of which was assessed using behavioral assays to determine the differences between wild-type mice and genetically modified mice that were either treated or not treated. For his work in the Gal Lab, Trendov presented at the 2024 Society for Neuroscience Conference and the Wayne State University's 2023 Summer Undergraduate Research Program Symposium, Translational Neuroscience Program Symposium and Research Day, Warriors Scholars Conference for Undergraduate Research and Creative Work. Prior to the Gal Lab in 2022, Trendov worked in the Healing Hydrocephalus Lab of Dr. Carolyn Harris and directly worked with PhD candidate Ahmad Faryami to assess shunt resistance and analyze the degrees of obstruction for shunts removed from patients who had undergone revision surgeries for their hydrocephalus. Additionally, he has volunteer experience at the Huda Clinic and DMC Clinical Research, and during his undergraduate career was the vice chairman of event coordination for the WSU Neurosci chapter. And our second wonderful panelist is Chris Turner, who is a senior at University of Virginia, where he's anticipated to graduate in May 2025 with a BA in Neuroscience Distinguished Majors program. After graduating, he'll take a gap year to continue his research and prepare for medical school. Turner was awarded a Neurosci 2024 travel grant as well for his project Cellular and Synapse Level Characterization of the Wide Field Vertical Cell Circuitry in the tree shoe superior colliculus. Under the guidance of his current PI, Dr. Aleb Arizir?
Arashir.
Yes. Arashir. Okay. The Arashir lab studies corticotectal projections of the mouse and tree shoe utilizing viral neuroanatomical tracing, confocal microscope, and the Allen Brain Atlas Connectivity Atlas. He has previously researched with Dr. William Levy in the Levy Lab studying episodic memory creation through hierarchical reciprocal networks from the hippocampus to associative areas, and particularly focused on literature review and using the Allen Brain Atlas Gene Expression and Connectivity Atlas. For his research, he has presented at the Society for Neuroscience Annual 2024 Chicago Conference and the 2024 University of Virginia Foster's Symposium for Undergraduate Neuroscience. Turner has a wide range of leadership experiences, including working as an emergency medical technician for the Charlottesville Albemarle Rescue Squad, a men's leadership program mentor with the Women's Center at UVA, and a health unit coordinator in the neurointensive care unit at UVA Hospital, a Camp Kesem counselor, an applied behavioral technician, and a Gift of Life bone marrow registry campus ambassador. He is also the co-founder and co-president for UVA's Surgical Interest Group and has received the UVA Helping Hands Scholarship for outstanding academic achievement and service to others, the Alumni Student Cooperative Scholarship at UVA for broad intellectual interests and the qualities of leadership, merit, service, and citizenship to UVA and the Charlottesville community. And he's also an Eagle Scout. So thank you so much, Alexander and Chris, for joining us. Is there anything that you all would like to say about yourselves? I know that I kind of already gave a mouthful, but if you want to say anything, I'm happy to have you do so.
Honestly, I think you did a great job. That covers just about everything.
Yeah, they were good questions you asked. I think that covers the majority of stuff we do. But also in the neurosci clubs at our respective schools, of course, which is a great opportunity to get exposed to this type of thing.
100%.
Well, I'm really glad to have you both joining us, and to just kind of kick it off, I'd love to know what initially got you interested in neuroscience, and what was it about your schools that made you choose those programs over something else?
Alex, you want to go first?
Sure. Actually, it's a very unique story. When I initially was graduating high school and applying for university, I was considering my options, and Wayne State was one of the top choices for me because it was local and also it was very research heavy. So I had a little bit of experience shadowing at a Wayne State lab that involved some kind of like research on ischemic stroke, and it was somewhat mythical to me because their hypothesis was that like a certain wavelength of infrared light can mitigate damage from this type of stroke. And so I got a little bit of exposure prior to my application to my undergrad. And then on a I don't know if it was like an info session or some sort of like orientation about Wayne State. They were announcing the onset, like the beginning of a neuroscience degree. It was actually, I think, in its second year of conception at the time of my application. And, I learned a little bit more about it and decided to, take the risk. And turns out I absolutely loved it.
That's a great story. Glad that you got in like at the beginning of the program. That's super cool. I think the UVA program actually has been going since around 2010, so we have some experience here. But I initially was interested in neuroscience. I'd say like in middle school, actually, I had like really bad migraines and headaches as a kid, and that just made me interested in understanding like my own condition. And despite the fact that I don't really research headaches or injury or anything like necessarily clinical related currently in my research, I still just have that innate drive to understand the brain and like human physiology. And so that is where my drive for neuroscience comes in. And then the reason I chose UVA, I wanted it to stay local. I'm from Northern Virginia, but also UVA compared to other schools like I was accepted to had such great opportunities. It has the hospital here, and so if any of you guys are pre-meds, you might want to consider places that have opportunities like that, but also there's just large amounts of funding too. And so it's really helped being at a large institution like UVA because because of the diversity of labs and different experiences that you can be exposed to. And so my first year, I was not actually in a lab or doing much research at all. I was focused on other things. And then second year, I realized like I came here for this reason, like, I should probably focus. And so I actually sent emails to 30 different labs here at UVA, across the psychology, neurosurgery, sociology, even departments. So like shotgun at out those applications, and then three responses came back. Two people actually wanted me in the lab, and so if you want to get involved in research, you got to put your name out there. But the one that came back was my old lab, Dr. Levy, and I was with that lab for 2 years, and I loved the work I did. He had me do literature reviews to understand like the connectivity of memory systems throughout the hippocampus. And um, but after that, I wanted more like bench work related things. And so he pointed me to my current lab, which is the Era Shear lab. And we studied like sensory systems of the brain. So we use a particular animal, like Tara said, the tree shrew, because it's a little more developed than is the mouse. And so it's a better analog to study the visual system. And so currently, I really enjoy my research. I'm doing a thesis right now, but also supporting my grad student with work that he does. And so I'm having a lot of fun learning new techniques. But yep, that's kind of my story getting into research and choosing UVA.
Cool. Yeah, that's really interesting that you both kind of like were aware of research and kind of like introduced to that prior to undergrad, because that's not always the case. And so that's really cool that you kind of had that initial like little dip in before actually getting to your universities, and then blossomed while you were there. So that's really awesome and cool that you both stayed kind of local to where you were prior to college. So um would you like to describe a little bit more about what your lab studies? And I know, Alex, you're not in a lab currently, but if you want to kind of talk about the research that you have done in your labs and um, yeah.
Sure. I'll kind of start chronologically. So I already discussed a little bit about the first introduction to research in regards to the ischemic stroke. And mind you, I had no idea what I was doing at this time. I was just kind of like getting involved, somewhat of like a shadowing type of experience where I could get a feel for what research was really like, especially animal research. And then moving on a couple years later, once I was actually at Wayne State, and kind of like Chris, I was figuring out what I wanted to do in life and decided to actually get involved in the hydrocephalus lab, as you mentioned. And uh, once again, actually, this lab was expanding, and so they were moving to a completely new building from the previous one that they had in the College of, uh, or the first one was at a building called iBio, which is about like 10 minutes off campus. It's not super far, but the place that they were expanding into was actually the College of Engineering, which was, I want to say, like a 2-minute walk from where I was staying. And so, similar to Chris, I was, like sending out emails trying to figure out what sort of labs I want to get involved in. And this one actually struck me as very fascinating, and especially because it integrates not only neuroscience, but kind of like a biomedical engineering approach, which frankly isn't my field of study whatsoever, but I loved it. And when I started there, uh, it was really cool to see all the different majors that were working in the lab, both undergrads and also like PhD students that were involved. And just to tell you guys a little bit more about what that lab studied. So uh, I'll provide a bit of context and then move into that. So hydrocephalus is characterized by increased intracranial pressure in your brain. And so there's several things that can cause this. Some of them are chronic and long lasting, possibly due to a genetic condition, for instance. And for these patients, the main course of treatment is, I don't want to say barbaric, but it's a little wild. It involves like a shunt being directly inserted into the ventricles of the brain and then kind of being like wired down into the stomach for drainage. And so this allows like a normalization of the pressure, but being that this is a highly invasive neurosurgery, it poses some risks. On top of that, the main issue in hydrocephalus in today's day and age is the fact that patients that have undergone this like shunt invasive surgery, they tend to go, they tend to have to like repeat the surgery because one component of the system inevitably fails over time. So they have to like redo this once again, pretty dangerous procedure just to kind of normalize their symptoms. So um, what our lab was looking at that time was kind of a holistic approach on bettering hydrocephalus treatment. And so they were working on 3D printing different shunts that could be inserted, different kind of like coverings on the shunts that would prevent protein aggregation and whatnot, using things such as like 3D printing and also developing a way to simulate CSF flow. And so that's kind of like the big picture. So they were looking at like different components of the therapy overall. And my role in particular was, as you mentioned, Tara, kind of assessing like shunt resistance in those that have been removed from patients that have undergone these revision surgeries. So it was very special because um, this lab had access to a biobank that contained hundreds of shunts, and we would run these tests to see like whether or not this component was actually the issue or was it something later on with like the valve or whatnot. So that's kind of what I was involved in for several months. And then later on, I kind of moved on from the lab, focused on wrapping up my undergraduate. And in 2023, I was I was kind of missing the research experience, so to speak. So I applied for a summer undergraduate research program at Wayne State University, and was accepted into this like neurological lab that had a focus on animal models, kind of bringing it back to like what I started with. And so this lab, as you also mentioned, was it has a lot of different um, studies, so to speak. So they were looking at myelin, but also like demyelination disorders. And a, s- a rare subset of these demyelination disorders is the one that I was studying called Pelizaeus-Merzbacher disease, which is a mouthful, and it took me probably a month alone just to learn the pronunciation. But um, big picture, we essentially developed a mouse model that recapitulates the most prevalent genetic mutation in patients with PMD, and we were using a AAV adeno-associated viral injection to kind of like mitigate the symptoms of this mutation. And so my role in particular was a little bit of literature review at the beginning just to get oriented with the field and what's going on, but then transitioned more so into behavioral assays to kind of assess motor symptoms and determine whether our therapy was effective or
That's super cool that you are involved in research that is so translational. But now you're also looking at the more cellular side of these pathologies. I think that's a great way to experience the field, especially for a lot of pre-med folks here. But my experience through research, so like I said, I was in a lab for 2 years that was focusing on episodic memory formation via reciprocal networks through from the hippocampus to association areas. And so what that means is basically my professor, he was a theorist and he was trying to understand how we create episodic memories in novel environments and how when you enter a new environment, how the particular context that is present in this situation is different than past situations and how that affects your memory of like the spatial scene. And so what he had me do was do a large literature review of the connections stemming out of the hippocampus, and we tried to figure out the reciprocal connections from hierarchical regions, meaning a region of the brain that projects downwards from high order to low order, and then regions of the brain that project from low order to high order regions. And so that's particularly important because in sleep, um, your hippocampus consolidates memories, and so having those reciprocal connections may be able to potentiate the memories that you created during the day in those novel contexts. And so I learned a lot of methods in that lab, just using online tools that are widely available and you can play around with yourself. One being the Allen Brain Mouse Connectivity Atlas. It's a super cool resource. The Allen Brain Institute is a very well-funded organization in Washington State. They do thousands of experiments on different things all the time, and they have this database online of these thousands of experiments they've run that is open source, available for the public. And so I use that to look at the connectivity of these different regions because they do tracer experiments. And so a lot of my initial work in my undergraduate career had to do with the connections regarding memory formation in out of the hippocampus. And my professor, he actually he passed away last year, and he worked to like the very end, which is incredibly inspiring. And he was a major factor in my current success and my interest in neuroscience and research. But he was close friends with my current PI, who is Dr. Lev Ereshir, and they do similar work in that they look at like the sensory system connectivity essentially. And what I'm currently doing for my thesis is looking at cortical connectivity to the superior colliculus. And look, it's a comparative analysis across evolution, so that I'm looking at whether um, different cortical connectivity to the spirit colliculus might underlie specific visual spatial behaviors that species exhibit across evolution. And so I'm using four different animal models: the mouse, tree shrew, cat, and monkey. I'm not actually using the cat and monkey, but it's just a literature review for that. But I am doing experiments on the mouse and tree shrew looking at corticotectal connectivity. Um, But in the lab, we do a variety of things as well. We do the there are different cell types in the superior oh, sorry. If you're not familiar, the superior colliculus is a part of the secondary visual system, which underlies like visual, spatial, and reflexive behaviors in subconscious behaviors to like certain stimuli in your environment. And so we look at different parts of the superior colliculus and the cell types of the superior colliculus. Um, and we're trying to find ways to isolate those cell types using AAV viruses, which is a super cool translation, Alex.
Yeah.
We try to isolate and create cell type specific AAV viruses. Um, and look at the morphology and dendrite morphology of these specific cell types because there's so much we don't know about the breadth of cell types that exist in the brain. And so this is just one vital system that we've been looking at and is translatable to, um, if I were to say like any clinical research, it's like sensory deprivation or something like along those lines. But yeah, that's kind of my story. I also I want to present my for SFN last year I presented alongside my grad student, and then this year I am probably going to present my own research on my thesis that I'm completing this semester, which I'm really excited for. Um, but yeah, that's kind of my story through research.
Nice, very cool. Yeah, you both have like really cool experiences in research, and it's really interesting to see like kind of the different things that you've done. And sorry to hear about your former PI too. I'm sure that that was really impactful. And also it's cool to see how his memory kind of like lives on in you and the research that you got into afterwards. So
Absolutely, I appreciate that.
yeah. Um, so yeah, that's really interesting. And I think that it's interesting too how you both kind of like have a little bit of touches into like clinical populations and stuff, especially since you both are pre-med and like wanting to pursue med school. So it's cool to see like both sides of that. Uh, and I know that you both have had experiences working with like your PhD students or your grad students and stuff like that. So I kind of wanted to like just dive into that a little bit more because I feel like sometimes if you're not familiar with research, you may think that it's just like, your research assistants and then your PI. So can you kind of talk about what the structure of your lab looks like, and what it means to like work with a grad student versus like just kind of all the ins and outs of what that could look like?
Sure.
Yeah,
So in my most recent lab, the PMD or Dr. Gao's lab, at the time that I was researching there, we actually didn't have any grad students uh, a part of the lab, but in the hydrocephalus lab, we did. We worked very closely with a grad student, and actually, this like healing hydrocephalus lab, I'm I think they had like 3 or 4 grad students at the time, but the one that was transitioning over to the College of Engineering for more space for like 3D printing and things like that, um, I essentially worked with him as if he was a PI. So because he was like a third or fourth year PhD student at this time, um, he had like a very heavy focus on research at this point. And so, although I did meet the PI of the lab several times, I pretty much only interacted with the grad student on a daily basis when I would go in. And it was very interesting for me at that time because um, I previously didn't really understand the difference of like what a PI is or what a PhD student does. And my experience in the lab kind of fleshed that out. And a PhD student is typically going to run like a particular route of study. So, like the lab might have an overarching goal or mission, for instance, but the PhD is going to have like a particular subset of that. And that's usually what they're studying for their dissertation, which is kind of like the capstone project of a PhD. And if you guys don't know what a dissertation is, I might not be the best one to explain this, but to my understanding, it's like the culmination of all your work in a particular lab leads up to this moment at the end of your PhD where you're essentially defending your research findings and you'll present the study, first like explain it, and more in depth than you would do at like a normal conference or SFN, for instance. They really kind of like pick you apart and assess your understanding. But it's a very fascinating, but challenging hurdle that you kind of have to get over before you're granted your PhD. And so, uh, yeah, that's at least my experience. It was kind of like he would run the lab, he would um, decide this sort of experiment or that sort of experiment, or kind of like delegate their own um, either research assistants or people like me that would come in and just like try to learn more about the lab. So I think the best way, just to summarize, is they're kind of like a coordinator who's like running under the PI, essentially.
That's a great way to put it. I would definitely add on to that that every lab is different. My current lab has two PhD students, but we also have older research assistants that just work as like lab technicians. My the first lab I was in, I would just meet with my actual PI in his office every week for 2 hours, and we would discuss the literature that I had reviewed the week prior. That is a unique experience, but you can also have bench work experience, which is actually running experiments in the lab like a lab technician. And that, if you initially get into research early in undergrad, that would probably be run under a PhD student's supervision. Um, and then you could also do your own project, like which is what I'm kind of doing now, which is also under the supervision of a PhD student. But since I understand the methods a little bit more later in my undergrad career, I'm able to have a little more autonomy in the lab. But I'd say research is definitely a growth curve throughout college. You do not know what you're doing when you first get in. I did not. But you will get it. People like love to help you, but you have to ask questions and you have to put yourself in uncomfortable positions. You have to ask if I can present like a poster on this. You have to ask like what else is there to this particular pathway? Like, are there other questions to ask? There are always questions to ask. And the more things that come to your head, do not like let them just slip under the rug. Ask them to people in your lab, and because that might spur future opportunities for you later in your undergraduate career or graduate career. But I'd say people a lot like I went into college at least like thinking that I would study like Parkinson's disease because my grandmother had Parkinson's disease, or I would study like something very translatable, such as like headache, like prognosis or whatever, but I did not. You have to seek what's available and then make the most of situations, I'd say. Um, and so you can be dreamy, absolutely, early on, but then you also have to be practical and you have to find a lab and make the most of every opportunity that you have. And so um, at least I'd say my opportunities, I'm incredibly grateful for everyone like who's helped me. It's very hard to get into research without help. And so continue to thank everyone above you because it is like you're in other people's labs at the end of the day. And so it's, and they appreciate the work that you do, but you definitely have to reach out and put yourself out there to get experience and talk to these PhD students. But PhD students love helpers too. So it's kind of a win-win.
Yeah.
Yeah, well said. I completely agree with what you're saying. And the main thing that I want to highlight is don't be afraid to ask questions. They expect you not to know, especially when you're starting. Um, but they appreciate curiosity, they appreciate initiative. So if you can display that, especially on the onset, that you're curious and um, it's kind of like this paradox of you don't know how much you don't know until you kind of learn more about it. And that's really the reality of research. The more you get involved in it, you kind of see how deep the pond goes. And yeah, well said overall.
Yep.
Yeah, that was also a point that I was going to highlight was I think that that's really crucial is to like be curious and to like just actually ask the questions because you know, you're not going to be able to know unless you ask, especially if they are, you know, because everybody already has their own questions that they're kind of exploring and stuff and really like learning a technology or a process or the exploring future directions comes from that those like initial questions of learning and being uncomfortable to eventually get comfortable in some kinds of ways. Um, but yeah, I think that it's interesting that you both have had kind of like different experiences between both of the labs that you were in, of whether you were working directly with your PI or if you were working more with a PhD grad student, what that looked like. So, um, I kind of think that that like got into a little bit of what I want to ask next, which is what kind of like preconceived notions did you have about research before actually getting into it? And how have those been kind of supported or debunked or just changed along your research experience?
Yeah, I'd say that it's a long road. The preconceived notion is that you can answer a question with one specific experiment. Um, my old professor, I think one of his like favorite quotes was like he wants to do the right experiment, not the next experiment, in that there if you want to answer the question, like you can there are a million different experiments you could do, but maybe they're not the right ones, but he wants like to focus on the right ones. And so having yeah, so preconceived notion is that it's a long haul. So it's not as you're not gonna be able to get that satisfaction like in your first or second year or a few months in the lab. Like if you go into research, it's a long experience, and you're in for the long run to learn a lot about the research, but also yourself.
Yeah, I think that's a great point. And honestly, I'm frankly, I'm not sure if I had any preconceived notions of research because genuinely I didn't know much until I got into the lab. Um, I suppose one of the big things is that, before you get into a lab, the picture that was painted, at least in my mind, was that every lab is only doing benchtop like experiments where they're like pipetting things or doing like cellular analysis or um, assess lab rats or mice or other animals. And I would say that it's a lot broader than that, especially because research can either be kind of like in a lab setting or in the clinic. So I know we've been talking primarily about kind of like the lab setting, where you're doing like animal studies or cellular studies, but there's also the aspect of clinical research where it's kind of like the next step above, where you're talking to real people and um, either having like medical devices or oftentimes pharmaceuticals are being assessed for their efficacy in human populations. So I think that the big thing, um, at least for me, along with that is similar to what Chris was saying. It's a very long process, and genuinely, I had no idea when prior to starting in a lab how long it actually takes to get publishable results. Could you go on PubMed for your, I don't know, science class assignment to do whatever, or sometimes even a quick Google search and you see all the articles. But it's difficult to appreciate how much work actually went into publishing all of these articles, until you're in that research setting and you realize like, wow, they must have had to run X number of experiments over several years and kind of had to refine their study and their methodology to get to that point of publication. So Big picture, I think um, it's a lot broader than people may anticipate, and also it takes a lot more time.
Yeah, I think that those are great points, and especially the broadness, because something that I've been especially hearing about as I've talked to more folks in these kind of settings is that like, research isn't just limited to STEM, it's everywhere. And then also to what you were talking about, like traditionally, or not traditionally, but oftentimes it is thought that research is just bench work, but there is so many clinical applications and stuff. And I know that in my own experience, like that's primarily what I was doing. Like I was working with EEGs and fMRIs and like language analysis based stuff. So that was like, I don't know, that was kind of mind blowing for me. I was like, whoa, this is also research. And it like makes sense when you actually like read papers and stuff. But to see it in practice, it's like, oh, research is so many multitudes of experience. Um, So awesome. I know that you both have kind of talked a little bit and like touched a little bit on like getting to publications and stuff, but kind of a step before that is like presenting. So would you want to talk to us about your experience presenting at SFN? What went into that preparation? How was that experience of presenting when you were there? Um yeah, what was your preparation process like?
Um,
Feel free.
We so I like I said earlier, I presented with my grad student at this past SFN, and we presented together. But I would say presentations at conferences can be different things. They're large and they're small conferences. SFN is a massive conference. It had 20,000 people at it. And not everyone gives a talk and not everyone presents. You do different things. They're incredibly diverse. People go for different reasons. And so we just presented a poster, but many other grad students and many other PIs, they gave 5, 20, or even hour-long talks. And so there are many different things that you can do. I'm actually going to a conference this weekend. It's the Central Virginia Society for Neuroscience conference, but I'm giving a 5-minute talk at it, not a 20-minute talk. But I'm preparing for it actively right now. But the preparation for that includes creating a slide deck, making sure that your talk is scripted and the transitions are clean and it has a logical flow. It includes creating a poster board that is comprehensible. Um, and it includes making sure that your results are projected how you want them to be because you know everything that you know and like about your research, but no one else knows anything about it. And so you need to present it in a way that is digestible for the different audiences that you might interact with. And one more last piece of advice, actually, when you are presenting, if you guys begin to present in your undergraduate career, I would say definitely be confident. You know what you have done. And so when people ask you questions, just trust your instinct. You just trust what you have done and rely on your knowledge of the subject. But that entails you knowing your subject. And so it requires studying and requires really knowing your stuff. But if you've been doing it for months, you'll definitely get it. But yeah.
I agree with everything that you just said there. I think my big suggestion is that you can never prepare enough. Um, Even if it seems excessive at the time, going into your first presentation, I would say definitely dedicate ample time to practicing and refining your presentation and also what visual you're going to be presenting to an audience, whether it's a slideshow or whether it's a poster. Um, in regards to SFN, the majority of people that attend and present at SFN are poster presentations. And so just to put it in perspective, you walk into a massive hall and there's hundreds of people that have posters up on walls. Um, and so if you're one of the people that have a poster up on a wall, kind of put yourself in the shoes of somebody that's walking past all of this, right? So keep this in mind when you're designing your poster for a conference, especially SFN or any sort of large conference. You want to make it both visually appealing, but as Chris says, digestible. So if your poster just looks like a research article with tons of text or very incomprehensible language, then it tends to get overlooked, to be honest. So, um, at least for me, when I went to SFN, there was like a period of time when I was presenting, but then there was also a period of time where you could like leave your poster up for a little bit and walk around to see other people. And so keep that in mind. When you have your poster up there, but you're not there to present, make sure that if somebody is walking past and it catches their eye, that they can like stand there for a couple minutes, read it, and get the gist of what you are trying to convey to the audience. I would say that's like very massive. But in terms of actually presenting, um, my biggest thing, and I have to give a shout out to Dr. Tom Fisher, who is the director of neurosci at Wayne State University. Um, he told me very bluntly, you want to make your speech probably 2 minutes. I would say get through what you want to say in roughly 2 minutes, give or take a little bit, but you don't want to like take up 15 minutes of somebody's time going into the nitty gritty of every single detail of your study. You want to convey the information and you obviously don't want to gloss over things. So this is what takes a lot of practice because you'll have your poster or your slideshow, for instance, and especially if you've done a lot of research or learned a lot about this subject, you could probably talk about it for like half an hour, maybe even longer. But true skill is when you can kind of condense this information down in an understandable way and it's concise and it gets to the point that you're trying to make. Um, and most often than not, if you do a good enough job at that and the person is interested, they're going to sit there for a few more minutes and ask you questions. And that's like a great opportunity to delve a little bit deeper in. But make it concise, make it digestible, and make it look pretty. Everybody likes it pretty.
Yeah, I
Totally.
think that those are great tips. Both the confidence and the making sure that it's like digestible. And I think that that's a really awesome point, Alex, that I had never considered about like if somebody's just walking by and they look at your poster, like can they actually understand it without, you being there to like dive into more of the nuance that's available there. So yeah, I think those are really great tips and also good luck on your presentation this weekend. I'm sure that you'll do great.
Thanks a lot. Yeah.
Um, so to kind of like switch gears a little bit, um, what does a day in your life look like? Both in the lab, outside of the lab, Alex, for your master's classes. I think that that is maybe something that like maybe folks don't realize. So if you want to like talk a little bit more about that, but you
Sure.
also don't have to.
No, that's perfectly fine. So when I was actually in my lab, I'll first start with this and then kind of transition. So when I was starting in my lab, I was an undergraduate at the time. And so although I was taking more credits in my undergraduate than I am in my graduate courses, um, the load overall didn't feel as intense. And so because of that, I had a bit more time to kind of like dedicate to going to the lab. And so when I would go in, I believe at the beginning it was like every day during the summer because during the summer I wasn't taking any classes during this research program, so I got really invested at this time. And then throughout the semester, to kind of like balance coursework and other obligations, I went in weekly to conduct my experiments a couple days a week and do like my data analysis at home, for instance. So at that time, it was kind of like a balance in between classes and lab work. But as I graduated from my bachelor's degree or my undergraduate, um, I was planning on applying to medical school, and I had like a really unique decision to make. It was either take a gap year and then study for the MCAT, prepare for my application, apply, and then also have to wait a whole nother year to start medical school. Coincidentally, Wayne State actually offers a master's program that fits into that timeline kind of perfectly. So this master's that I'm involved in, it's the BMS program or Basic Medical Science, and it provides like an opportunity to sort of step into med school or like transition into med school a little bit more fluently. Um, the reason that this is frankly going from undergraduate courses to graduate level courses. I feel like I've developed a new appreciation for actually understanding the material on a deeper level. And this is very necessary in medical school and as you kind of continue on. So you have to really refine your study techniques and also your kind of your dedication to classes. For instance, in undergrad, you can kind of like push things off until later on, right before the exam, do a little bit of cramming and then probably do well. Whereas grad school, it's a whole different ballgame. You have to be on top of things constantly. And this is the exact same in medical school. Like you have to constantly be studying. I know Chris probably knows a lot about this because he did memory systems, but you have to constantly like refresh and reconsolidate those memories. So I think that this master's kind of like offered me that uh, learning opportunity to to figure figure out my studying and overall balance in life. But on a day to day for me, in like now, um, I'm still taking master's classes, but also studying for the MCAT, and so that's why I discontinued my time in the lab. Frankly, it was just to make sure that everything went well on this application cycle. So I'm not really like postponing it later on. So 3 days a week, I go to classes on campus and they run for a few hours. But then afterwards, I tend to go to the medical school library and do some studying, some overall refreshment of the content that I either just learned or learned throughout the week. Um, and then coupled to that, I'm I'm like about a month or so out from the MCAT, so it's really like grind time, um, nose down and spending as much time as I can dedicating to that. So that's sort of my day to day, and also layering in a little bit of clinical work. I don't want to take up too much of the time, but it's kind of like classes, MCATing, clinical work, and getting my ducks in order for the med school applications, essentially.
I actually have a similar type day to day to Alex currently. I also have my MCAT in a month, so good luck to you as well, Alex. We're in this
Thank
together.
you, likewise.
But yeah, the MCATs take up a lot of my time right now. That's I attribute about 4 to 5 hours a day to that currently. I'm sure you can relate, Alex. It's tiring, but I also, of course, so current typical day is class about 2 hours a day, and then lab for about 3 to 4 hours. Different projects could be helping my grad students project or finishing up my thesis, and so that could include like data analysis of the past experiments, writing methods, writing the intro, things like that. I have to finish up my thesis and then go home and study for MCAT for about 4 to 5 hours. So usually jam-packed days, but trying to make the most of my last um, push here in college and just focus and leave and do my gap year on a good note. So yeah, it's my day to day.
Nice. Well, good luck to both of you. I'm sure that you will do amazing. So I am not even worried about that. But
Thank you.
um, yeah, good luck. Um, yeah, and kind of as we finish up our last couple minutes here, I just wanted to know if what advice do you have for new neuroscientists in general and for those who are looking to get into research?
I would say that neuroscience is a very exciting field and will continue to be so over the next several years. So I would say appreciate that there's still a lot of progress to be made in the field of neuro, whether it's neuroscience, neurology in general. Um, and I think that really provides us a unique opportunity, um, which is another reason why I actually really fell in love with this field genuinely. I would say, as far as practical advice, don't be afraid to put yourself out there. Chris had an excellent story of how he was applying to several labs before he heard responses. And I would say that that's the norm. So don't give up. Don't send it out to like three people. And then if you don't hear back from them, be like, oh, shoot, maybe this isn't for me. Continue at it. And I think that your efforts will inevitably be rewarded at the end. When you find the lab, you start getting involved and you learn more. And I think that it's like a snowball effect. Once you get in a research lab, then you kind of like gain some experience, and then you might be a little bit more attractive for other labs that are looking to hire people in, or um, especially unpaid undergraduates. Oh my God, they would love that. But on a serious note, I would say put yourself out there. Don't be afraid to learn. Be curious and pursue your passions. And I think when you couple those things together, um, your dreams can really come true, as cliche as that sounds.
Yeah, that's awesome. I totally agree. My final advice, I guess, would be you drive your own ship. You really got to make it happen for yourself. People want to help you, but you got to put yourself in a position where they can help you. And so that means actually going to the lab. If you have the option, go to lab. Or if you have the option to send an email, which you do, send the email and put your name out there. But also present yourself in a way that shows your skills and shows that you're capable of doing certain things. And so being being a utility to the lab helps. And so like try to learn things and like uh give people your attention, your time of day so that you show that you're available to learn. And so yeah, you drive your own ship.
Nice. Yeah, I think that those are great pieces of advice. Yeah, being like, you drive your own ship and be curious and put yourself out there. I think that those are all great. And I think that you both have given a lot of like insight and like little nuggets of tips and advice and stuff throughout us talking. And since we're right at 8 o'clock here, I will close out and say thank you so much for chatting with me and for sharing your perspectives and everything. Um, yeah, how if there's anything else that you all would like to say before we kind of switch into a more open and casual conversation, feel free.
Let's say one last thing. Chris, you actually reminded me. At least for my university, which is a very research heavy university, but especially in any university that you may be going to. Um, if you're looking for somewhere to start, I would say the best opportunity is just speaking with your professors. A lot of times, a quick Google search of your professor will pop up like their profile on whatever your school's website is. And more often than not, it'll show you what their field of research is. So let's say you're a young neuroscientist and you're looking to get into research, but you have no idea like where do I look? I would say first look at your professors. And even if they don't have any availability in their lab, they might be able to point you in the right direction. So I would say that they're a great conduit and they're there to help you. If you show the interest and you show that you're willing to participate, um, good things can happen, essentially. But overall, thank you for the opportunity to speak today. It was an honor to meet you guys and looking forward to keeping in touch, hopefully.
Yeah, definitely. I second that. If you guys are here, you're definitely motivated individuals and good young neuroscientists. So keep it up.
Whoa. Switch out of structure and formalities. Chris, feel free to, head out and do what you need to do.
Yeah.
And we'll be here. So do
Perfect.
what you
I'll
need to
try
do.
to head that. Thank
Yeah,
you.
sounds good. Yeah, thank you.